Hello, my name's Rosine Niyibikora. I'm a student of Ines Ruhengeri in Department of BLS Biomedical Laboratory Science in Faculty of Health. I'm going to present about assignment of applied immunology assignment, where my task is from question 31 to question 35. Question 1 is talking about what is principle behind agglutination. First of all, let me start by defining what is agglutination reaction. This reaction is immunological technique based on specific interaction between antigen and the antibody. The reaction resulting in the formation of visible clumps or aggregates. The principle behind this agglutination reaction is occur when soluble antibodies bind to particular antigen. Vice versa means particular antigen binding to antibody to form a close-linked complex. The reaction depends on three key factors. One factor is specificity, second is close linking, third one is lattice formation. About specificity, each antibody recognizes and binds to unique epitope of antigen. Then for close linking, when antibody bind to antigen, they form what we call close linking between particles. This network of connection results in visible clamp is what we call agglutination. For lattice formation, it occurs when multivariant antigen interact with bivariant or multivariant antibody, where they create stable lattice structure. This visible network confirms presence of target antigen or target antibody. This agglutination occurs in four types. First type is direct agglutination, where antibody react direct with antigen on the surface of cells. Example, red blood cells, bacteria. And this direct agglutination is used in blood grouping, bacterial identification, and others. And second one is indirect agglutination. In indirect agglutination, soluble antigen or antibody are attached to innate carrier such as latex bed or charcoal particles. The coated particle agglutinate in the presence of target analyte. Example is detection of rheumatoid factor in autoimmune disease and is used in rapid streptococcal antigen test. Third one is reverse agglutination. In this reverse antibody, instead of antigen, are attached to innate carriers. This antibody-coated particle agglutinate when they encounter the target soluble antigens. This reverse is used in detection of bacterial toxin and hormones. The fourth one is hemagglutination. In hemagglutination, red blood cells serve as carrier for antigen or antibodies. The reaction involves clamping of RBCs, which are red blood cells. This hemagglutination is used in blood typing, detection of virus like influenza, hemagglutination inhibition test. So this agglutination is applied in blood grouping like ABO and Leisers grouping, is applied in infectious disease diagnosis like detecting bacteria and viral infection such as streptococcal infections and this salmonellosis. and is also applied in a pregnancy test where it detects hormone called human chorionic gonadotropin, were produced during pregnancy using latex agglutination assay, and also is used in autoimmune disease diagnosis where it identifies autoantibodies against self-antigen, aiding in the diagnosis of conditions like rheumatoid arthritis or systemic lupus or erythematosis, and also is applied in virology where hemagglutination assay help detect viruses that bind to red blood cells such as influenza and rubella. Allow me to go on second question, which is asking about how does latex agglutination work in diagnostics. First of all, latex agglutination is diagnostic technique that leverage or use specific interaction between antigen and antibody to detect presence of disease markers such as pathogens, immunoresponse in patient sample. The procedure, how do we perform the latex agglutination? First of all, we prepare latex bed. In the first step, latex beds are coated with either antibodies or coated by antigen. This coating ensures that the latex particle can specifically bind to target molecule in the sample. Second one is mixing with patient sample. The coated latex bed are mixed with the patient sample, which can be blood, urine, cerebrospinal fluid, or other body fluid. If the target antigen or antibody is present in the sample, it will bind to the corresponding counterpart on the latex bed. Third procedure is formation of antigen-antibody complex. If the sample contains the target molecule, it will bind to coated latex bed and forming close-linked network of bed and annulates. This interaction creates visible agglutination, cramping of the latex particles. The fourth procedure is visual detection. The agglutination is typically visible to naked eye or under microscope, where a positive result is indicated by formation of clumps in suspension, while negative results show a smooth or uniform solution without visible crumbs. This latex agglutination has like three types. It has direct latex agglutination, indirect latex agglutination, and reverse latex agglutination. In the direct latex agglutination is where used to detect antigen in the patient sample. For example, detecting bacterial antigen in cerebrospinal fluid to diagnose bacterial meningitis. Second one is indirect or passive latex agglutination. This type is where designed to detect antibody in the sample. For the direct, we detect antigen in the sample, while for indirect, we detect antibodies in the sample. For instance, it is used in diagnosing autoimmune disease like rheumatoid arthritis by detecting presence of rheumatoid factor. For the third one is reverse latex agglutination. Here, latex beds are coated with antibody to detect soluble antigen such as toxin or protein. This latex agglutination is applied in infectious disease detection and is applied in autoimmune disease diagnosing, also applied in inflammatory markers, where to detect markers like C-reactive protein, which indicate inflammation or infection, and also is applied in the pregnant test, where it detects human chorionic gonadotropin in urine. Allow me to go on third question, which is asking about what is the principle of precipitation reaction in immunology. Let me start by precipitation reaction. Immunology definition is based on specific interaction between a soluble antigen and soluble antibody to form insoluble antigen antibody complex. This complex aggregate into large structure that precipitate out of solution and also create visible or measurable precipitation. Mechanism of precipitation reaction. The first one is antigen antibody binding. When soluble antigen and antibody are mixed, they bind specifically to form antigen antibody complex. Both antigen and antibody must be multivalent, means having multiple binding sites to allow close linking between them. The second one is lattice formation. In lattice formation, precipitant occur when antigen and antibody must be in optimal ratio to create a stable lattice network. This network grows larger, eventually becoming insoluble and forming visible precipitation. The optimal ratio is found in the zone of equivalence where antigen and antibody concentration are balanced. This precipitation reaction has like three types: in solution, in gel-based precipitation or immunodiffusion, and immunoelectrophoresis. Application in diagnostic, where we apply precipitation reaction. Detection is applied in detection of specific antigen or antibody. Precipitation reaction are used to diagnose infection by detecting antibody against specific pathogen or identify antigen from bacteria or viral source, and also is applied in quantification of immunoglobulin. Here is a technique like radioimmunodiffusion, measure the level of immunoglobulin like IgG, IgA, and IgM in a patient's serum, aiding in diagnosing of immune deficiency. Also is applied in identification of protein, where immunoelectrophoresis help identify abnormal protein and assisting in diagnosing conditions like multiple myeloma or autoimmune disease. Let me go to fourth question. Is asking about how does the radio immunodiffusion test performed. First of all, radioimmunodiffusion is quantitative immunological technique used to measure the concentration of specific antigen, typically protein, in a sample. And it is commonly used in clinical laboratories to quantify serum protein such as immunoglobulin, IgG, IgA, and IgM, and complement components. Principle behind that radioimmunodiffusion is that radioimmunodiffusion is based on a precipitation reaction between the soluble antigen and the antibody. This principle is when antigen diffuse radially from a well into a gel containing specific antibody, it forms a precipitation ring where the antigen and antibody reach the zone of equivalence, means the optimal antigen and antibody ratio. The diameter of this ring is directly proportional to the antigen concentration in the sample. By comparing the ring size to standard curve created from known antigen concentration, the unknown antigen concentration can be accurately determined. How do we perform this test? First of all, we prepare gel plate. How do we prepare it? Incorporating antibodies, where we have agarose gel is prepared with specific antibody evenly disturbed throughout. Then the gel is poured into a petri dish or in a glass plate and allowed to solidify. Secondly, we create well. Well is a small circular well, 3 to 4 millimeter in diameter, are carefully punched into the solidified gel. This well will hold the antigen sample and stands. Second procedure is loading the sample. Standard solution. Known concentration of target antigen are added to separate well. These standards serve as reference for constructing a standard curve. And also an unknown sample in patient or test sample are added to remain a well. Third one is incubation. That plate is placed in humidified chamber at an optimum temperature, which is 25 to 37 degrees Celsius for 18 to 48 hours. During this time, the antigen diffuses radially from the well into the gel and interacting with the antibody. The fourth one is formation of precipitation rings. As the antigen diffuses outward, it binds with the antibody in that gel to form insoluble antigen-antibody complex, where a visible precipitation link appears around each well at a zone of equivalence. The fifth one is measurement and quantification, where after incubation, the diameter of each precipitation ring is measured using a ruler or caliper. A standard curve is created by plotting the diameter of ring, where y-axis, which is y-axis, and against a known antigen concentration on x-axis. The antigen concentration is an unknown sample is determined by comparing their ring diameter to this curve. This is applied in quantification of serum protein and is applied in detection of complement protein and also applied in monitoring immune disorders, also in quality control in biopharmaceutical. Let me go to fifth question, which is last one. Is asking about what is principle of ELISA technique. First of all, we have to understand what is ELISA technique. ELISA in full word is enzyme-linked immunosorbent assay, which is used in the immunological method for detecting and quantifying specific antigen or antibody in biological samples. And it combines the specificity of antigen-antibody interaction with the sensitivity of enzyme substrate reaction, making it powerful tool in diagnostic and research. Principle behind that ELISA technique. This ELISA rely on the specific bind between antigen and antibody to detect the presence of target molecule. The key features of ELISA is the use of enzyme conjugated antibody, where this antibody is where we add that enzyme on antibody. The enzyme combined with antibody is called antibody conjugated or antigen, which upon reaction with a specific substrate, that substrate is a chromogen which give a color when there is presence or absence of that target antibody or antigen. Procedure proceed a color change or other measurable signal. The intensity of this signal is proportional to concentration of target molecule in that sample, where that allowing for both qualitative and quantitative analysis. How do we perform ELISA technique? First of all, we cutting the microplate. This microplate example when a well of microplate are coated with either antigen for detecting antibody or is coated with antibody for detecting antigen. Secondly, we block. In blocking, non-specific binding sites are blocked using blocking buffer, example, bovine serum, albumin, or keratin, to prevent false positive results. Thirdly is sample addition. The sample containing the target molecule, which are antigen or antibody, is added to well and incubated to allow specific binding, means to allow reaction between antigen and the antibody. Fourth is detection antibody addition. An enzyme conjugated detection antibody is added, which bind specifically to target molecule or antigen antibody complex. Fifth one is substrate addition. We add substrate for getting a color. This substrate is taken as a chromogen. After washing away an bound component for antigen antibody complex, substrate specific to the enzyme is added. The enzyme catalyzes a reaction that generates a detectable signals, usually a color change. Six is signal measurement. The signal is measured using a spectrophotometer or microplate radar. The intensity of color change is proportional to the concentration of target molecules. Then we go to interpret results. This ELISA is working in three types. First one is indirect ELISA. This indirect ELISA is where antigen is immobilized and primary antibody bind to it. An enzyme linked secondary antibody then bind to primary antibody. This indirect, it has advantage of high sensitivity due to signal amplification, and it is applied for detecting specific antibody in serum such as HIV test. Second one is sandwich ELISA. In this sandwich ELISA, it used two antibodies where for capture and the detection antibody. For the target antigen, the capture antibody immobilize the antigen and detect the antibodies conjugated with enzyme binding to antigen. In this sandwich, we use two types of antibody where we use antibody for detecting and we use conjugated antibody which is combined with that enzyme. Then for competitive is where we use word coated with antigen while we are detecting also same antigen. The sample antigen compared with label antigen for binding to limited number of antibody. Higher antigen concentration result in weak signals. This ELISA is applied in clinical diagnostic, where we detect infection disease like in HIV, hepatitis, COVID-19, autoimmune disorder, and hormonal levels like in pregnancy test. Also is used in vaccine development, where we measure immune response by detecting antibodies against a specific pathogen. And also is applied in food and environmental health, where we detect allergen, toxin, and contaminant in food and environmental samples. And also ELISA is used in research for quantifies protein, cytokines, and other biomarkers in biological samples, aiding in understanding disease mechanism and immune response. Here is the end of my presentation. Thank you for kind and attention. Thank you for watching my video. God bless you.